Hi, my name is Alessandro Lodesani. I'm one of the co-founders of the Quantum Biology Institute. Today I'm going to tell you a little bit about my work at the Institute, especially when it comes to instrumentation. But before we do that, I want to tell you why we chose to work on what we're working on, which is how weak magnetic fields impact biology. So we decided to study this, first of all, because we do think that this interaction is mediated by quantum effects, which are called spin superpositions. And a big reason is that all of this investigation can lead to a new knob to actuate on biology, which is, of course, magnetic fields. The magnetic fields that are needed to control biology are pretty weak. And this means that it's very easy to produce them. And this puts us in a very special position where knowledge is immediately translated into application and devices. Because when we decipher which magnetic field does what in which organism, then creating a device to apply that magnetic field becomes very, very easy, because the engineering behind creating magnetic fields is very well established. The only thing that we're missing is the knowledge of what magnetic field we need to use. There's lots and lots of evidence that magnetic fields affect many different biological processes. So this has the potential of impacting a bunch of different technological fields, such as anti-aging, health, theranostics, food security, space travel, and even quantum technologies, and learning from nature how to engineer better quantum technologies. Our specific goals at the Quantum Biology Institute are to gain a comprehensive understanding of how magnetic fields impact biology on many different levels, starting from the quantum states of electron spins inside of proteins that undergo chemical reactions, all the way to physiology and everything in between. We also want to demonstrate that the interaction between magnetic fields and biology is mediated by quantum effects, namely these spin superpositions. Once we do that, we also want to understand how to control these spins and how the control of these spins translates into biological effects. This way, we're gonna get predictive power on how to use magnetic fields to actuate on biology. Right now, at the Quantum Biology Institute, we have 4 different levels of investigation for how magnetic fields impact biology. The first one is the quantum level. So basically what we want to do is to look at the fluorescence intensity of proteins and how that changes with magnetic fields at the nanosecond timescale with single photon resolution. So this will be the undeniable proof that the interaction is mediated by quantum effects. And what we want to see basically is an oscillation in the fluorescence intensity, and we want to prove that we can tune the oscillation by changing the magnetic field environment. The second level of investigation is proteins. So we want to study and map which proteins are sensitive to magnetic fields in a way consistent with the quantum model, and we want to understand the molecular features that are necessary to have this kind of sensitivity. Then we have a nomics approach, which is a more top-down approach, basically looking at the biological processes and how those are affected by magnetic field. Lastly, we are starting right now an investigation on metabolism and reactive oxygen species and how those are affected by magnetic fields. Hopefully, one day we'll put all of these together. You can probably tell that these 4 different levels are very different. They require very different specialization, but they all have one thing in common, which they require new instrumentation. To this point, most of the people that do biological experiments just don't pay attention to magnetic fields. We do believe that magnetic fields have a large impact on biology, and so what my job is developing instruments that have magnetic field control so that we can perform all of these experiments and keep track closely of the magnetic field environment. Right now, I'm going to tell you about the instrumentation plan and the progress on it at the Quantum Biology Institute, where basically we want to study magnetic field effects in biology. Let's dive deep into the instrumentation. I created a classification of the instruments that we're building at the Quantum Biology Institute, and I divide them into 3 classes: long-term exposure to magnetic fields, real-time response, and our quantum microscope, which is basically going to combine the 2 spectra. So what I mean by long-term exposure is we perform experiments where we pick our biological sample, we place it in a magnetic field environment that we can control tightly for a certain amount of time, then we take it out and measure it. And we can do this with many different magnetic field conditions, and we can compare between those. Then what I mean by real-time field response is that we can keep track of the fluorescence of proteins and how that changes by changing the magnetic field in real time. Then our quantum microscope is going to be able to do both at the same time in the same experiment. So it's going to keep track of the fluorescence and look at it with single photon resolution at nanosecond timescales, looking at how the spins of the electrons are behaving as a function of the magnetic field. But it's also gonna basically keep track of the biological outcome over a long exposure to a certain magnetic field that triggers the spins to behave in a certain way. And so this basically closes the gap from the quantum to physiology. Now, let's take a closer look at the instruments that have been developed for long-term field exposure. This is our hypermagnetic chamber that we can use to go from the Earth's magnetic field and below, all the way to about 1 nanotesla, which is a field that is even lower than what you find in outer space. And again, the idea of the long-term exposure is that we place our samples inside of the hypermagnetic chamber and leave it there for a bunch of time, and we sort of look at the chronic exposure to hypermagnetic fields. These are coils that I built that can produce a very, very uniform field, and the idea is just you place your sample inside and we can control the field from about the Earth's magnetic field to around 30 millitesla. The cool thing about this is that you can tightly control temperature by using water cooling, and we can do sham experiments as an additional control. And then we have the magnetic rings, which are the cheapest and easiest way of applying a uniform magnetic field, but these have a fixed magnetic field value, so you cannot change it. Now let's take a look at the instruments that can do real-time field response. So this is a plate reader. It's an instrument that is commonly used in biology for high throughput screening. The way this is built, there's not much room for magnetic field control. So the kind of magnetic field control that we can have is going to be relatively poor in terms of uniformity and how tightly we can control the magnetic field. But this can be a very good screening platform for detecting if there is any change in the fluorescence of your proteins as a function of the magnetic field. And once you see something, then you can move to other instruments to get more precise data. This is our bacterioscope, which we can use to keep track of the fluorescence of bacterial plates with a camera. And we can change the magnetic field in automated experiments using this electromagnet. You can actually take a look at some of the data that these can produce in our new publication. There. And this is a very, very good screening platform for looking at magnetofluorescence in bacterial plates. But the problem that this has is that it can go only from the Earth's magnetic field and up in terms of intensity. And so it can go up to about 200, 300 millitesla, but it cannot go below Earth's magnetic field. And this is why we have the hyperbacterioscope. So I am currently building this, and basically this is just another version of the bacterioscope, but instead of going from the Earth's magnetic field and up, this goes from the Earth's magnetic field and down. And by combining the bacterioscope with the hypobacterioscope, we can get a full spectrum of how protein fluorescence behaves as a function of magnetic fields. The next step after the hypobacterioscope is completed is gonna be to create a quantum version of the bacterioscopes. And so the concept is going to be very similar, but instead of keeping track of the fluorescence over long periods of time, we want to look at how the fluorescence behaves at the nanosecond timescale with single photon resolution. And these will basically tell us information on how the spins behave in a coherent way. The final piece of the puzzle is going to be our quantum microscope, which basically does all the things that the quantum bacterioscope can do, but can also keep track of biological imaging. It will be able to look at the spin states of electrons inside of proteins that undergo chemical reaction and link this behavior all the way to physiology by fluorescence microscopy. We think that the experiments that are going to come out of this are going to be crucial for the growth of quantum biology, because these correlate quantum states to biological outcomes. And performing these experiments a bunch of times with different magnetic fields will basically give us what we're calling the code book on how to actuate on biology with magnetic fields.